Welcome to G Fun Facts Online. Today we are abandoning the human scale. We are shrinking down to the level of the undergrowth, beneath the bark of rotting logs and into the silken architecture of the canopy. We are here to witness a silent war. It is a conflict of microscopic proportions but epic complexity, a Russian nesting doll of biological interactions where the very concept of an individual dissolves into a colony. We often view arachnids, spiders, scorpions, ticks, and mites, as solitary creatures. We see them as lone predators or pests. We see the wolf spider hunting in the grass or the orb weaver waiting patiently in its web. But if you look closer, through the lens of high magnification macroecology, these creatures reveal themselves to be not just hunters, but landscapes. They are mobile ecosystems. They are walking biological islands that support complex communities of residents. Some of these residents are distinct hitchhikers, merely using the arachnid as a bus. Others are thieves, stealing food from the host's table. And some are masters of physiological hijacking, rewriting the genetic and behavioral code of their hosts to serve their own ends. But the complexity does not stop there. These parasites have their own parasites, known as hyperparasites. And those hyperparasites may carry symbiotic bacteria that alter the entire chain of command. This is the world of Hyper-Symbiosis, a theoretical framework for understanding the multi-trophic, recursive, and often bizarre networks of life that exist entirely on, within, and around the arachnid form. To understand hypersymbiosis, we must first reconceptualize the host. Consider a large female golden silk orb weaver of the genus Trichonephila. To us, she is a spider, but to the thousands of organisms that interact with her, she is a geological feature. Her web is a city, and her body is a continent. The web of a large spider is a resource-rich environment that attracts far more than just prey. It attracts argyrodes, also known as dewdrop spiders. These tiny, silver-bodied relatives of the black widow are obligate kleptoparasites. They live in the webs of larger spiders, moving stealthily across the non-sticky threads to steal wrapped prey bundles, cut silk lines to consume the protein, and even eat the host's egg sacs. In the arid southwestern United States, the relationship between Argyrodes Pluto and its host, the black widow, reveals the first layer of this parasitic ecosystem. The argyrodes are not just nuisances. They represent a significant tax on the host's energy. But here's where it gets interesting. The argyrodes themselves are not immune. They are prone to their own set of predators and parasites, creating a microfood web entirely suspended in the air, independent of the ground below. The host spider's web becomes a battleground where the owner is often the least active participant, merely providing the infrastructure for a bustling economy of theft. Now, let's zoom in further, onto the cuticle of the spider itself. This is a jagged terrain of hairs, spines, and chitinous plates. Here, the mitescape flourishes. Mites are the most diverse biological hitchhikers on Earth, and arachnids are their preferred vehicles. This phenomenon is called foracy, where one organism uses another for transport. But in arachnids, it reaches absurd levels of complexity. A harvestman, or daddy longlegs, wandering the forest floor, may carry larval erythra-eid mites attached to its legs. These larvae are parasitic, drilling into the harvestman's leg to suck hemolymph, or spider blood. However, they are temporary residents. Once engorged, they drop off to molt. The fossil record gives us a window into how ancient this war is. In Baltic amber dating back 50 million years, we find evidence of this relationship frozen in time. A whirligig mite, a predator in its own right, was found with a planidium attached to it. A planidium is the first instar larva of a small-headed fly from the family Acroceridae. This is a rare glimpse into a chain that goes parasite on a predator on a potential parasite. Acrocerid flies are typically parasitoids of spiders. The fact that one attached to a mite suggests a case of mistaken identity, or perhaps a complex multi-stage life cycle, where the mite acts as a bridge to get the fly larva closer to its true spider host. The most dramatic examples of hypersymbiosis occur when the parasite does not merely eat the host, but becomes the host, commandeering its nervous system. We call these the physiological hijackers. The polysphincta group of ichneummonid wasps are the premier bioengineers of the spider world. A female wasp finds a spider, temporarily paralyzes it, and glues an egg to its abdomen. The spider recovers and continues its life, weaving webs and catching prey, completely unaware that the larva on its back is slowly draining its fluids. For weeks, the relationship seems commensal, almost benign, but as the wasp larva prepares to pupate, it injects a chemical cocktail, likely ecdysteroid-mimicking hormones, into the spider. This triggers a specific, genetically dormant behavioral program. The spider stops building its normal capture web and instead constructs a cocoon web. This is a reinforced, minimalistic structure designed solely to protect the wasp pupa. Once the web is finished, the spider sits in the center, and the larva consumes it entirely, leaving only a husk. The larva then spins its cocoon in the safety of the web the spider was forced to build. This is what biologist Richard Dawkins calls the extended phenotype in action. The wasp's genes are expressing themselves through the spider's behavior. If the wasp is a horror movie monster, the fungus Gibellula is something far stranger. Gibellula is a genus of pathogenic fungi specialized to hunt spiders. Unlike generalist fungi that simply kill and consume, Gibellula manipulates the spider. An infected jumping spider or cellar spider will be compelled to leave its hiding spot and climb to the underside of a leaf or a high cavern wall, a location optimal for spore dispersal. There, the spider bites down, locking itself in place, and dies. The fungus then erupts from the spider's body, not as a shapeless mold, but as structured, colorful stalks that often mimic the appearance of an insect, potentially luring other predators to their doom. But here enters the hyperparasite. Recent research has uncovered that Gibellula itself is often victim to another fungus, Penicillium thielii. This is a fungus that does not infect spiders directly. It infects the Gibellula that is infecting the spider. In this hyperfungal war, the Penicillium overgrows the Gibellula fruiting bodies, stealing the nutrients the Gibellula stole from the spider. Imagine the layers here. Level 1 is the spider, the base resource. Level 2 is the Gibellula, the zombie maker controlling the spider. Level 3 is the Penicillium, the thief consuming the zombie maker. From an ecological perspective, the Penicillium might actually be beneficial to the spider population by suppressing the virulent Gibellula, acting as a natural check on the zombie plague. Let us move to the recursive nightmare of ticks and mites. In the world of ticks, blood is the currency of life. But obtaining it is dangerous. Some ticks have found a hack: hyperparasitism, or feeding on their own kind. In colonies of the soft tick Ornithodorus, scientists have observed a behavior termed homovampirism. Adult males or hungry nymphs will attach not to a mammal, but to a fully engorged female of their own species. They pierce her cuticle and drink the blood she has already processed. This is more than just cannibalism, it is a vector pathway. If the female tick carries spirochetes bacteria, like Borrelia, which causes relapsing fever, the hyperparasitic tick can acquire the pathogen directly from her, bypassing the vertebrate host entirely. This creates a closed-loop transmission cycle, where the disease circulates within the tick population, independent of the external world. We also see this recursion in the concept of mites on mites. Pseudoscorpions, tiny, scorpion-like arachnids lacking tails, are famous for foracy. They grab the legs of giant beetles or flies to hitch a ride to a new habitat. However, these pseudoscorpions are often carrying their own passengers, smaller mites that hide in the folds of their exoskeleton. And within the gut of those mites, bacteria. A specific example of this multi-layered transport occurs with the pseudoscorpion Nanowithius warhamani. It lives in symbiosis with harvester ants, but to disperse, it has been recorded hitchhiking on a scorpion. Here we have a pseudoscorpion, riding a scorpion, likely carrying mites, all moving together through the desert night. However, not all hypersymbiotic relationships are parasitic. Some are surprisingly cooperative, forming invisible alliances. Deep in the Amazon rainforest, the massive Colombian lesser black tarantula shares its burrow with a tiny humming creature, the dotted humming frog. In a normal scenario, a tarantula of this size would view a frog as a snack. But this tarantula has a specific recognition code for this frog. It allows the frog to live in its burrow, even crawling under the spider's massive fangs for shelter. In return, the frog acts as a specialized pest control agent. Tarantulas are plagued by small invertebrates, ants, mites, and parasitic flies that attack their eggs and spiderlings. The massive spider is too clumsy to catch these tiny invaders. The frog, however, specializes in eating ants and flies. The spider protects the frog from snakes and large predators, and the frog protects the spider's lineage from micro-predators. This is a mutualistic symbiosis, embedded within a world of parasitism. We see similar cooperation internally. Scorpions are ancient survivors, and part of their success may be due to their internal microbiomes. Recent genomic studies on scorpions like Vajovis smithi have revealed specific lineages of mycoplasma bacteria living in their guts, and surprisingly, their stingers. Unlike pathogenic mycoplasma in humans, these scorpion symbionts likely play a role in nitrogen recycling, allowing scorpions to survive in nutrient-poor deserts, or even in the synthesis of venom precursors. The scorpion is not just a stinging animal, it is a walking fermentation tank, reliant on its bacterial passengers to survive. The existence of these hypersymbiotic networks drives evolution at a breakneck pace. This is the Red Queen hypothesis in overdrive. Every organism must constantly evolve just to stay in the same place. Spiders evolve tougher cuticles to stop mites. Mites evolve sharper chelicerae. Spiders evolve unpredictable behavior to confuse wasps. Wasps evolve more potent neurotoxins to override that behavior. Furthermore, the pressure of parasitism drives the evolution of sociality. Some theories suggest that social spiders, who build communal webs, evolved sociality, partly to form a guard caste that can remove parasites from the colony, a collective immune system. And when we look at speciation, the process multiplies. When a host spider splits into two species, its parasite often splits too. But in hyper-symbiosis, a split in the spider forces a split in the wasp, which forces a split in the wasp parasitizing wasp. This creates a cascade of speciation, generating biodiversity at an exponential rate. To study hyper-symbiosis is to realize that there is no such thing as a solitary organism. An arachnid is a node in a vast invisible network. When a tarantula walks across the forest floor, it is a mobile city. It carries phoretic mites on its legs. It houses mutualistic frogs in its home. It fights off parasitoid flies that want to turn it into food. It hosts gut bacteria that help it digest that food. And potentially, it carries fungal spores that are waiting for the right trigger to turn it into a zombie. We must stop looking at the arachnid and start looking into the ecosystem it represents. In the end, the arachnid world is not just about 8 legs and venom. It is about the millions of years of negotiations, thefts, alliances, and hijackings that have allowed these creatures and their riders to conquer the earth. Thanks for listening to G Fun Facts Online.